안녕하세요. 저는 토론토 대학교의 약학대학에서 박사과정을 하고 있는 박엘리아입니다. 저희 실험실에서는 폴리머와 지질을 나노 단위로 이용해서 뇌와 뇌로 약을 효과적으로 전달하는 방법을 연구합니다. 오늘 발표하는 내용은 그런 특수한 나노 약물 전달 방식을 알츠하이머병 치료에 도입해 더 효과적인 약효를 볼 수 있다는 것을 증명한 예시입니다. 오늘 연구는 어드밴스 사이언스 학술지에 이번 연도 2월에 게재가 되었는데요. 관심이 있으면 찾아보시길 바랍니다. 발표 시작하겠습니다. Alzheimer's disease is a chronic and progressive neurodegenerative disorder leading to cognitive dysfunction, causing 60 to 70% of cases of dementia. So the diagnosis of the disease is based on the neuropsychological tests with the help of neuroimaging, such as PET, CT, MRI, that can detect the morphological changes in the brain. Currently available treatment consists of acetylcholinesterase inhibitors or NMDA receptor that can modify the neurotransmitter to reduce the symptom of the disease. So there's been a recent approval by FDA as a disease-modifying drug, which is aducanumab and lenacanumab, but their effect still needs to be validated and actually being questioned as well. So there's actually practically no disease-modifying treatment available for the AD up to date. So the reason for this is primarily because of the complex pathology of the disease. It is well known that the toxic proteins accumulation, such as amyloid or tau, play important roles in the progression of the disease. However, the research has found out that it's not the only cause of the disease, but only a part of the whole story, like you see in the figure here. So there's other pathology that are involved in the disease, such as synaptic dysfunction, inflammation, oxidative stress, and vascular dysfunction that actually interplay each other and multiply other factors to make the disease even grow bigger. So even if you, for example, reduce the vascular dysfunction, it may not be enough to stop disease or even slow down the disease because the other disease factors are not treated and left untouched. So there is a need for development of the disease modifying agent that can target the multiple disease factors. Reactive oxygen species and hypoxia play important role in the Alzheimer's disease for many reasons. So amyloid beta, which is known to be the main factor for the disease, can increase the reactive oxygen species, which react with the cellular components and make the cell dysfunctional. And also amyloid beta form plaques that get accumulated onto vessels that will reduce the blood flow by blocking the vessels or make the vessel size smaller. That will lead to reduction in the oxygen level in the cellular level, which is called hypoxia. And oxidative stress can lead to oxidative damage in the cells that will lead to cell death, which is called apoptosis. And oxidative damage can, of course, increase the neuroinflammation in the brain. And lastly, oxidative stress can activate the angiogenesis, which is a new vessel formation by activating VEGF, which would lead to abnormal vascular and lymphatic structure in the Alzheimer's disease. So in summary, reactive oxygen species and hypoxia are very important in the progression of and the development of Alzheimer's disease by initiating or multiplying the other disease factors that are involved in the disease. In addition to the complex pathology of the disease, there's one more challenge that we have to take into consideration in treating the Alzheimer's disease, which is common for any CNS disease. So it's the existence of blood-brain barrier. So blood-brain barrier is a barrier that is located between the blood and then brain. So it sort of protects any foreign substances coming to the brain so that the brain is secure. While it has a protective function against anything that's potentially harmful, it actually poses us a challenge in delivering any drugs into the brain to treat the disease. So there has been some researches to deliver the drug into the brain despite the existence of BBB. The first thing is you can bypass the BBB by directly injecting the drug into the brain, or you can use the nasal administration to bypass the BBB. Or you can sort of modify the drug structure so that it can penetrate the cells or through the tight junction between the cells over here. But it can limit the ability of the drugs because you have to fit certain characteristic to be able to deliver the drug through this way. And then the next one is a BBB disrupting, which is temporarily disrupting the blood-brain barrier so that the drug can enter the blood-brain barrier during that temporary time. And then after a certain time, then the BBB will get healed into a normal structure. And the last one is receptor-mediated transcytosis, which is basically using a receptor that is expressed on the blood-brain barrier. So the blood-brain barrier has several receptors that allow only selective components to enter the brain from the blood. So for example, the brain still needs some glucose or insulin or anything that is needed for the brain function to cross the BBB to enter the brain. So if we make the drug that looks like food to the receptor, then the receptor will bind to this food or ligand looking drug and then transport this drug across the BBB so that we can deliver the drug safely into the brain. So in our lab, we have developed the polymer lipid hybrid nanoparticles that uses this receptor-mediated transcytosis to deliver the drug into the brain. So as you can see from this picture of the animals, you can see after administering into the intravenous vein, you'll see the drugs in the brain after 15 minutes, and the signal is remain there and slowly releasing the drug over 2 hours. So the technology uses the terpolymer, which has a starch backbone that is conjugated with polymethacrylic acid, which has a functional group that is attaching with the polysolvate 80. So polysolvate 80 can attract the lipoprotein in the blood so that these particles surround itself with the lipoprotein so that it looks like a lipoprotein, and then the receptor will transfer this nanoparticle into the brain. By using this technology, our lab has loaded different kinds of materials such as inorganic substances or the protein or even the gene delivery. So like I mentioned, the problem for developing the AD treatment is first primarily complex pathology of the disease, that single targeted therapy is not working that well to reverse the disease. And secondly, difficulty delivering the drug into the brain because of the existence of blood-brain barrier. So our approach is to use this platform to load the drug that has the multifunctional effect so that we have more effective therapeutic outcomes. This particle is called ABTP-MDNP. So AB stands for amyloid beta antibody that can actually find and target the disease area, and then which is attached to the terpolymer that I mentioned. And the terpolymer gives the particle the ability to cross the blood-brain barrier and being delivered to the brain. And in this vehicle, we loaded the manganese dioxide, which has the ability to reduce the oxidative stress and increase the oxygen in the surrounding system. So by having these effects is known to have antioxidative and anti-inflammatory effects, which were shown in the cancer models. So by combining these two approaches, we hope that this will provide better therapeutic option for treating the Alzheimer's disease. So the objective of this study is to evaluate the therapeutic effects of ABDP and DNP in the brains of Alzheimer's disease by reducing first hypoxia and oxidative stress, secondly inflammation in the brain, third vascular dysfunction, fourth lymphatic dysfunction, and lastly the progression of the disease, which is examined by the cognitive function and amyloid beta accumulation. For this study, we used a transgenic mouse model of Alzheimer's disease, which is TGCR-ND8, and we provided the treatment twice weekly for 2 weeks. And then we measured the biomarkers using immunohistochemistry and ELISA. And we also did some functional assay using MRI to measure the vascular function. And lastly, we performed the behavior tests in these animals as well. Firstly, the direct effect of nanoparticle is reducing oxidative stress and hypoxia, which was the first thing we did when starting this project. So as you see over here, CA9 is a marker for hypoxia, meaning if there's more expression, there is a low oxygen in the tissue. You see that in Alzheimer's disease brain, there is significant hypoxia happening in both cortex and hippocampus, which are the main disease region for these Alzheimer's disease. On the other hand, when we treat them with nanoparticle, we see a significant reduction in these markers. As you see, the color has been sort of fainted. And then after that, we did the oxidative stress marker examination in protein, lipid, and DNA. We found a reduction in the oxidative stress in both protein and lipid, but not up to the DNA level, which might be much deeper than the protein and lipid expression itself. And the total amount of reactive oxygen species measured by DHE was decreased by the nanoparticle treatment. Since nanoparticle was shown, the manganese dioxide nanoparticle is known to have some immune modulating function, we hypothesized that this particle can also reduce the inflammation. So we measure the inflammatory cytokines, which play important role in progression of the disease by making amyloid beta plaques more, or tau, or making the microenvironment a little bit more diseased. As we can see here, alpha-1 ACT, interleukins or TNF alpha were significantly reduced by the nanoparticle treatment. Similarly, we have measured the activation of immune cells in the brain, which are microglia and astrocytes. So similar to what we saw in the cytokines, the activation of these immune cells have been significantly reduced, which is good for treating Alzheimer's disease. So initially, inflammation may play positive role by helping with the clearance of the amyloid beta or tau. However, as it gets chronic, it even damages the surrounding tissues and is not able to properly clear out the amyloid beta from the brain, despite the chronic inflammation. So decreasing the inflammation play a positive role in treatment of Alzheimer's disease. Reactive oxygen species and hypoxia play roles in vascular function through the VEGF. So VEGF is angiogenic factor that gets activated by increased oxidative stress or hypoxia through the HIF-1 alpha. By reducing those factors, we have shown that in the Alzheimer's disease brain, there's reduction in the VEGF with the nanoparticle treatment. So this suggests that the reduction in VEGF can stop the chronic angiogenesis that's happening in Alzheimer's disease brain that actually makes the vascular function even more normal and not properly conveying any blood flow. So this can normalize the vascular structure in the brain so that it's more functional and being able to help with the neuronal health. So based on this observation, we went ahead and measured the vascular function using the MRI technique in these animals. First thing we did was to measure the blood flow in the brain. As we can see here, so this is wild type. Pre is before the treatment. Vehicle is after the treatment, which is 3 weeks apart from the pre. And then nanoparticle treatment is shown here. So we can see that after nanoparticle treatment, there is an increase in the cerebral blood flow in cortex and also deeper level of subcortex where hippocampus is. So improved blood flow in the brain is very important in a way that it can provide better nutrition and oxygen supply. And in addition, it can also improve the clearance of amyloid or tau that is toxic to the brain. So by improving the clearance and providing more food and oxygen to the brain, it can provide the better environment for the neurons, which should lead to improved cognitive function in Alzheimer's disease brain. Similarly, we measure the blood-brain barrier permeability using the MRI. And blood-brain barrier permeability can be dysfunctional because of the chronic inflammation that is happening in the brain, which makes it more inflammatory by damaging the BBB because there are some foreign substances that can enter, otherwise it wouldn't come in. So we found that by reducing oxidative stress and inflammation, we were able to the nanoparticle was able to reduce the BBB permeability, meaning that the integrity of the barrier gets better. Brain has a lymphatic system that is known to play important roles in clearing out the amyloid beta by conveying the cerebrospinal fluid. So you see this diagram, the green line is the lymphatic vessels in the brain. And this is more directly connected to the cells so that it can clear out the amyloid beta in more efficient fashion. So since it's also regulated by the VEGF, since the lymphatic is also regulated by VEGF-C, we expected that by reducing the VEGF, we will see a similar effect in the lymphatic function that we saw from the vascular function. So the first thing we had to do was to see if actually the particles are delivered to the lymph nodes in the brain. If not, it would not actually work. So we injected the drugs and then collected these deep cervical lymph nodes to see if the particle is found in these lymph nodes. And we found that ABTP-MDMP was successfully entering the lymphatic nodes in the brain in both wild type and Alzheimer's disease mice. So based on this observation, we went ahead to measure the lymphatic vessels in the brain, and we found the significant improvement in the structure. As you can see from the picture, it has more linear lines, while the untreated one was not really showing any clear lymphatic vessels in the brain. So based on this structural improvement, we expected the lymphatic function would be improved, which can be measured by the amyloid beta clearance in the CSF. So we measured the amyloid beta in the cerebral spinal fluid, and we found that there is an increase in amyloid beta clearance after the nanoparticle treatment in advanced stage of Alzheimer's disease, but in moderate stage, there was no significant difference, may suggesting that at moderate stage of Alzheimer's disease, lymphatic structure may not be that dysfunctional for us to be able to treat the disease. Based on our previous observations that I showed you just now, that the treatment with nanoparticle can increase the clearance of amyloid beta from the brain by vascular and lymphatic routes, we measure the accumulation of amyloid beta in the brain after nanoparticle treatment. So in line with improved amyloid beta clearance, the nanoparticle treatment was able to successfully reduce the amyloid plaques from the brain in both hippocampus and cortex. So you see these green dots are amyloid plaques, and the nanoparticle was able to reduce these plaques, possibly or partially from improved clearance of amyloid beta. Lastly and finally, we measure the cognitive ability in these animals after nanoparticle treatment. So for these tests, unlike other examination that we did, we treated them for 8 weeks, which on the other hand, the animals were treated for 2 weeks for the remaining experiments to make sure that we are seeing a positive effect if there is. So we use the fear conditioning test for this assay, and I can explain briefly about the fear conditioning test we used. So on day one, you provide the electrical shock with a sound in a certain square box to make the mouse correlate the shock with the sound and the box that the animals are. And on day two, you don't give any shock, but put the animals into the same box that it was exposed on day one. If the animals remember the correlation with the shock and the box, then the animals will react to it. That gives us the hint that the animals remember this and have a good memory of it. And on day three, we put the animal into a completely different box, and we give a same sound that we gave on day one. So with the sound, if the animal gets frightened, that means the animal is remembering the incidents. So these tests test different domains of the cognitive ability, contextual and acute, which is related to different regions of the brain. So our experiment showed that the nanoparticle is able to increase the cognitive ability in these animals in both contextual and acute memory, possibly due to its multifunctional effect, unlike other treatment that is only being able to treat certain domain or ability of the brain function. So in conclusion, our experiments show that ABTP and DMP, the nanoparticle, reduced oxidative stress, inflammation, and improved the blood flow and lymphatic function in Alzheimer's disease, therefore was able to reduce the amyloid beta accumulation and improve the cognitive function in Alzheimer's disease. I'd like to thank the funding sponsor for this study and your attention as well. 감사합니다.